Hello, welcome to Chapter 20. This is going to be a discussion of DNA tools and biotechnology. This is split into a three-part series, this one being Part 1. Now, let's talk about an overview of really the main techniques and some applications of biotechnology. So they can be split into two things. Let's talk about the techniques first. Some techniques that scientists use in the field of biotechnology include DNA sequencing, and that's pretty self-explanatory. You're determining the sequence of bases in DNA segments. Gene cloning. That's basically allows scientists to study genes once they've made certain organisms produce those genes. It's not ethical to use humans in those types of studies, so we have other model organisms. PCR amplification. PCR stands for polymerase chain reaction, and it's really amplification of segments of genes. And what amplification means is a significant increase in the number of copies of that gene or that segment of DNA. Now, in order to perform many of these other techniques, PCR amplification must be completed first. So we'll talk about that process, but a simple way to put it is basically DNA replication in a tube. Now, expressing genes, of course, organisms express genes naturally, but you can force organisms to express genes more so. You can knock out genes or basically make them not express a gene. And you can put genes in organisms that don't normally have that gene and have them express it and see the effects of it. And then once you have organisms express the genes, you might want to analyze that gene expression. So in order to see what's been expressed and how much has been expressed, or whether it has been expressed at all, you want to analyze it. And there's different methods to be able to detect the presence of a gene being expressed. And you can edit genes, right? So, there are natural mutations, but you can force changes in genes. And if there's a mutation that's detrimental to, let's say, humans, it causes disease or something, we've got methods now that we're investigating that allow us to edit those genes so that they become more normal and they do not cause disease. Now let's look at the applications. So applications include those applications in agriculture, basically genetically modified organisms, to determine the ancestry of individuals and animals, other animals as well. In medicine, right? So we've talked about some of these. We might want to express genes and see what's causing a particular disease and perhaps edit those genes to prevent the cause of the disease. And forensics, right? So to analyze genomes to determine the identity of an individual. And environmental cleanup. So a specific example of that might be, you might genetically modify an organism so that they can help break down oils that might have been spilled in the ocean. All right, so these are just some examples. And remember, the techniques are the process of doing things, and the application is what these techniques can be used for. So we're going to talk about DNA sequencing first and cloning of genes. And these are great tools for the process of genetic engineering and to inquire about biological genes, right? So what we want to do is maybe investigate the sequence of a gene and to perhaps predict its protein structure and predict its ability to have an effect on the cell. So DNA technology includes those tools which involve the manipulation of DNA. Now, it's not only DNA sequencing. DNA cloning would be a type of manipulation. Nucleic acid hybridization. So these types of techniques take advantage of the base pairing properties of DNA sequences. So remember, they're attracted to one another. The bases are attracted to one another by hydrogen bonding. And that's really what hybridization is referring to, is that attraction by hydrogen bonding. And you can use this to be able to detect certain sequences in DNA and RNA. Genetic engineering is the manipulation of genes for a practical purpose. So, one example that I gave in agriculture, genetically modified organisms. So you may want to genetically modify, let's say, rice. Rice isn't normally nutritious, but if you can genetically modify it so that inside the rice cells, it produces more vitamins. A lot of third world countries depend on rice for a food source. So, it'd be useful to make it a little bit more nutritious. Let's take a look at one method of DNA sequencing. This is called DNA chain termination sequencing. And again, Fred Sanger developed this. This is a very old method of DNA sequencing. It's very laborious and it takes a long time. But ultimately, the narrator spoke of a chemically altered DNA bases, and they're separated in four separate tubes. So one tube will have the adenine, one guanine, one cytosine, and one thymine. And again, they're chemically altered. So when they are incorporated into the DNA sequence, it stops the synthesis of the DNA because they're altered in such a way where the 3' hydroxyl group, which is actually required for the addition of the next nucleotide, it's missing. So that's what actually terminates the DNA sequencing. Now let's look at another method of DNA sequencing. There are newer methods that are considered what's called next generation sequencing. And these are techniques that are much more rapid and much less expensive and laborious than the chain termination sequencing that you just saw or learned about. So most of them, the basis of it is that the sequencing is done as the DNA is being synthesized, or sequencing by synthesis type of method. So first, the fragments must be copied. And we'll talk about this method later, but they're usually copied so that there are billions of copies of it so they can be easily detected. And they're copied by the process of polymerase chain reaction, or PCR. Once you have billions of copies of it, what happens is a single strand of each fragment is going to be immobilized. So how they're copied is that they have, during the process of PCR, a immobilization type of element is going to be added to the fragments that's being copied. So that's how you have that. Now we have something that allows us to immobilize the fragments. And so the single strands are going to be immobilized and they're going to be separated so that you have one strand of DNA sequence that's immobilized. And then the synthesis occurs. So then the complementary strand is going to be synthesized. So in the copying of the fragments, initially, you are actually synthesizing DNA, but then you're separating those strands, and then you're synthesizing it again in a different instrument. So the complementary strand is going to be synthesized, and the synthesis is being detected at the same time. So you can synthesize 1,000 bases to 100,000 base fragments, and it synthesizes up to 300 nucleotides in length. Right, so you can only by these methods, you can only detect up to 300 nucleotides long. But it can be sequenced in parallel, and you can combine all of the fragments to determine a full sequence. So this is an example of a high throughput technology. It can be sequenced in parallel. You can have 96 well plates, so you have 96 different samples running at the same time. Now, a sample output would be this right here, and it's a direct sequencing output that you can see. So we have these nucleotides across the top, which is what the instrument is going to call out. And this is the incorporation of a guanine. And the peak is relative to the amount of guanines that are present adjacent to one another. So this peak, this size peak represents one guanine. This represents one cytosine. And then there's no thymine right here. And then we have one adenine, and then we have two guanines in a row, and then two cytosines in a row, and one thymine. So this represents the DNA sequence. It goes G, C, A, G, G, C, C, T. This is a method that is used in several different types of technologies and instruments. I used a method called pyrosequencing, which is considered a high throughput technology, but it's one of the different variations of this type of sequencing. Now let's look at a method or a variation of this type of DNA sequencing that your textbook discusses. So here we have cells, and the genomic DNA is going to be extracted from the cell, and then the genomic DNA is going to be fragmented. Each fragment is going to be isolated with a bead. So using PCR, you can amplify the fragments, right? So we have here an example of 10 to the 6. So this is millions of copies of this fragment. They're going to be made, and as they're made, a bead is attached at the 5' end. So the bead is going to be placed into a well. With these, during this process, the bead is going to be placed in a well with DNA polymerases and primers. So, of course, the DNA polymerases and the primers are going to be used to amplify the DNA or synthesize the DNA. So here we have inside the well, we have the bead, the DNA sequences, and all of the DNA polymerases and the primers. And the DNA sequences are going to be serving as the template strand. So these are DNA sequences that have been extracted from the cell, and then they're going to be placed into these wells right here. So there's going to be solutions containing each of the 4 nucleotides. They're going to be added to all the wells with this DNA. So this is an example, a close-up of a single well. So you can see you can do this all in parallel. You can have very large trays with many different wells. So I used, in the pyrosequencing method, I used 96 well plates, but there are actually plates that have much larger amounts of different wells on there. So the solutions containing the nucleotides, of course, the nucleotides are going to participate in the DNA sequencing. So the DNA polymerase is going to incorporate the nucleotides in order to copy the strands of DNA that are in each of the wells. So all of this is going to be kind of washed off, right? So all the excess stuff then is going to be washed off, and then this process is going to be repeated within all of the wells. So this kind of shows the process here. So on the bead, we have these DNA sequences. The DNA polymerase is going to attach to the single strand of DNA sequence, and it's going to start incorporating bases to copy the DNA. So now it's going to make single strand pieces of DNA into double strand pieces of DNA. So it's DNA synthesis. So the process of this is going to involve the production of light. And now there's other enzymes in there that allow the production of light. But basically, if a nucleotide is incorporated, the release of pyrophosphate, which is the two phosphates, right? So one phosphate is going to stay to the DNA strand. The other two are going to be released. So inorganic pyrophosphate is going to be released, and this causes a flash of light. And this flash of light is detected by the instrument. And you saw that picture that I showed you before, right? So it detects that, and the larger flash of light is going to represent more than one nucleotide of that same nucleotide being incorporated. So then you get a larger peak. So as we look at this process, if a nucleotide is not complementary and it's not incorporated, then there's going to be no release of pyrophosphate. So then there's going to be no flash of light. And that's where you saw that gap with the adenine in that picture before was not, there was no peak present. So the process is going to be repeated until every fragment has been completely synthesized, right? So copied in this case. And of course, all of the flashes of light are going to be recorded by the instrument. And you can see here, this is very similar to what you saw on the previous slide. This is just an example of a pyrogram that is due to one of the new methods. So here we have a thymine. Here we have a cytosine, and then there was nothing incorporated here, so it goes thymine, cytosine, and then adenine, and then guanine. And then we have cytosine and then guanine, so you can see the key up here. I'm just using that. Now, guanine and then thymine, and then we have two adenines, and then we have two guanines, and then we have one thymine, and so on and so forth. So it can detect up to four to five in a row. After that, it's not as accurate. All right, so that's basically the basis of the sequencing by synthesis methods, and there's different variations of that. So like I said, pyrosequencing, and then this is kind of like pyrosequencing, but it's a more high throughput method, which has been developed based on the pyrosequencing type of. Now, a little bit more about DNA sequencing. There's third generation sequencing techniques that are even faster and even less expensive that have been developed. So in this case, we could have one DNA molecule that can be sequencing as it moved through a small pore. This is called nanopore DNA sequencing, and this pore is going to be present in a particular membrane. So in this case, each base interrupts an electric current differently. So the difference in the electric current is going to be measured, and that tells you what base is going to be incorporated. So here we can see we have the membrane right here, and we have a set of proteins right here, and we have the DNA sequence that's going through. This is the pore. The DNA is going through the pore, and then we have DNA polymerase synthesizing the DNA as it's passing through the. Now let's talk about copying DNA sequences. So I already kind of discussed the process of PCR, but we're going to go through that process a little bit more in detail and describe it. And then we're going to move into, let's say you want a copy of gene and then you want to clone it in order to study it. So let's talk about making multiple copies of a gene or just any DNA segment, right? So these are processes that allow you to do that. So DNA scientists can prepare DNA segments. So they can extract DNA from cells and prepare segments and create millions of copies of this DNA. And you can do it by cloning. In this case, with cloning, plasmids are used. So we've learned about plasmids. Remember, they are small circular pieces of DNA. They're extra chromosomal that are often in, bacteria and in archaea. And they're generally not necessary for the organism to survive, but these plasmids have been used in order to clone genes into other organisms, including bacteria and eukaryotes. Now, the plasmids have been modified from the natural plasmids that are found in bacteria, in general, right? So they've been discovered and now they're being used in an application. So the plasmids are used in the application of DNA cloning. So in this case, an overview of this, and we'll see a picture of it, which kind of gives you a little better idea what's happening. But DNA, your desired piece of DNA that you want to study, it could be a DNA segment or it could be a particular gene that you're looking at. It's inserted into the plasmid. And this allows the creation of a recombinant DNA molecule. So what recombinant is basically combining two sources of DNA. Now we have this recombinant DNA molecule. It's a recombinant plasmid. So it's the original plasmid with your insert in it. Now let's look at this process. So if we look here to the left, here we have a bacteria. This is a natural bacteria with its chromosome and then the extra chromosomal plasmid. So we can imagine that we have plasmids. You could just buy the plasmid. You don't have to actually extract it from the bacterium. But this is ultimately where the plasmid comes from. It comes from a bacterium, which originally made it naturally or transferred it by horizontal gene transfer between bacteria and archaea. But now we actually use those in the lab for this application. Now we have the plasmid, and the plasmid ultimately has been modified so that it has what's called the multiple cloning sites, and it has what's called restriction sites, which allow us to cut the plasmid open so that we open the plasmid and then we can take a gene and put it into the plasmid. So basically it's a cut and paste type of mechanism. And there's a lot that happens in between this process right here. So obtaining the gene from the cell of interest, right? Or the gene of interest from the cell and the type of organism. And this is the gene. And now you must copy it and then you can put it into the plasmid. So this is what creates a recombinant DNA plasmid. So after that, we put it back into bacteria. So it's probably not going to be the same bacteria that it originally came from, but it could be, right? So we could have plasmids that we're actually storing in bacterium, and then we're going to extract it from the bacterium, and then we're going to insert the gene of interest, and then we're going to put it back into a culture of bacterium. So this is going to be considered the recombinant bacterium. So in this case, this bacteria is going to have its original plasmid, its original chromosome, and within the original plasmid, there is going to be a gene of interest. So this is a transgene. It comes from another organism. So these bacteria are going to be grown in a culture, so they're fed all the nutrients that they need to so that they can replicate. So then they're going to replicate, and now you're going to have not just two, right? So this just shows one step of one generation of a replication, but they're going to be replicated so that there are millions of copies of these bacteria. So now we have millions of organisms, each containing the plasmid, which has been copied during the process of fission. Now, since your gene of interest has been inserted into the plasmid, and now you have it inside bacteria that are actually copying this, and the genes are being expressed. So in order to survive, these genes are being expressed, and you can direct the gene of interest to be expressed as well. So let's assume that this gene of interest is being expressed in E. coli now. So this could be a gene from a eukaryote like us, and we can insert that gene into E. coli and make E. coli produce that protein. So we could use this protein or modify bacteria in ways which can be applied to certain problems that we have. So it could be used for basic research and other types of bacteria or of applications. So we can put a gene for pest resistance and insert it into plants so that they're resistant against, animals that might, insects and other animals that might affect their growth. We could use the gene which alters a bacteria for cleaning up toxic waste. So I explained that on the first slide of this segment. So this is one way in which it can be done. The protein can also be harvested and then used. So for example, human growth hormone, it's actually expressed and overexpressed so that we can extract it and use it as treatment for diseases. Insulin is another example. And then we could have proteins. We can overproduce proteins that can dissolve blood clots. And it can be a type of therapy. So these are just examples that are used in medicine. Now let's watch a little animation about this process.
We can use the techniques of DNA technology to recombine and copy genes. A restriction enzyme is used to cut open a plasmid, a small circular DNA molecule obtained from a bacterium. The plasmid serves as a cloning vector. The restriction enzyme cuts only at a certain DNA base sequence, called the restriction site. The same enzyme is used to cut up DNA obtained from a human cell. The restriction enzyme produces DNA fragments that have matching sticky ends that join by complementary base pairs. An enzyme called DNA ligase forges covalent bonds that join human and plasmid DNA, creating recombinant DNA.
All right, so that kind of shows the process of creating the recombinant DNA using a plasmid. So basically inserting a gene into a plasmid, and then you can do with the plasmid whatever you want. All right, so let's talk about how multiple copies of a gene or any other DNA segment are made. So I've been kind of touching on this, but now let's look at the actual process a little bit more in detail. So gene cloning can allow you to make multiple copies of a gene. So we just shown that process, right? So you can have the copy of the gene that is inside the plasmid, inside bacteria, and then make the bacteria grow. So that's an indirect form of replicating a gene or increasing the copies of a gene. So in this case, the plasmid is used. It's a foreign gene, which is going to be inserted into a vector. And these types of vectors are called cloning vectors. So there are different types of vectors, and they're actually plasmids. So there's plasmids that are cloning vectors. They serve as vectors, which is like a vehicle to allow things to stay or be moved around. In this case, we have a cloning vector, which is meant to replicate fragments of DNA and store these fragments inside bacteria. Now, there's also expression vectors, which are meant to express whatever gene has been inserted into the vector. Right, so a cloning vector is just basic. It's meant to just store this and replicate a copy of DNA. So these types of plasmids are readily obtained, easily manipulated, and they can be easily introduced into bacterial cells. And of course, bacteria can replicate very fast. E. coli can replicate as little as double, every 20 minutes if you provide them the right conditions and enough food. Now, for practical purposes, of course, we want to amplify genes in order to make more copies of it, ultimately to produce protein products, which can be used for research and in medical and other purposes, like basic research studying the protein. I've done that. So the animation described restriction enzymes and kind of told you what they do, but they're used to make recombinant DNA plasmids. So let's talk about that real quick and what a restriction enzyme is. So restriction enzymes have been discovered in bacteria, and what they do is cut at what's called restriction sites to create restriction fragments. So restriction enzymes, they're restriction enzymes because they cut at specific DNA sequence sites. So they're areas in a chromosome or a piece of DNA that they recognize these certain DNA sequences, and then they cut the double-stranded DNA. Now, the most useful ones are going to cut it in a staggered way, leaving an overlap. These overlaps are called sticky ends. The sticky ends basically bond with complementary sticky ends, and this allows for the gene to be incorporated, or can allow for the gene to be incorporated in a certain direction. And the sticky end, the enzymes that create sticky ends, create the sticky ends, and the sticky ends are make it a lot easier to put the insert into the plasmid. Once the plasmid is inserted into once the plasmid and the gene of interest or the segment of interest is connected by complementary base pairing, by using the same restriction enzymes, DNA ligase is used to basically seal those bonds. So there's little gaps that are made, and you probably remember this from General Biology 1. I've posted some old videos for you to review if you don't remember this, but ultimately during DNA replication, there are on the lagging strand, there's fragments that are made, the Okazaki fragments, but the fragments must be connected, and they can't be connected in the 5' or the 3' to 5' direction. So it leaves gaps. And DNA ligase naturally comes in and connects those bases that are adjacent to one another. Well, this must be used in research too, in a tube, right? So DNA ligase seals these bonds, or basically fills the gaps and connects the adjacent bases so that the insert is actually part of the plasmid now. It's fully connected into a circular form of chromosome. This allows the research, of course, to join the fragments from the different sources. So now we have this single fragment, which is the recombinant DNA plasmid.
Restriction
Now let's
enzymes
look
are
at this
enzymes
process in a
isolated
little
from
animation.
bacteria that recognize specific sequences in DNA and then cut the DNA to produce fragments called restriction fragments. Different restriction enzymes recognize and cut different sequences of DNA. This restriction enzyme recognizes the DNA sequence GAATTC. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence AATT. This restriction enzyme recognizes the DNA sequence CTGCAG. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence ACGT. This restriction enzyme recognizes the DNA sequence CCCGGG. It cuts the DNA strands between the bases C and G. The resulting DNA fragments have no single-stranded sticky ends.
So ending at this, we have a fragment or two fragments that have been created which have no sticky ends. These are called blunt ends. The blunt ends can make it very difficult to insert a fragment of interest or your gene of interest into a plasmid. However, it's used in some cases. All right, so let's look at this process in just a picture format. Okay, so we have a bacterial plasmid, which has multiple restriction sites, and I mentioned there's a place where you can use, it's designed or been changed from the original bacterial plasmid so that there are multiple restriction sites adjacent to one another. So you can choose a restriction enzyme. Sometimes you want to pick, well, you definitely want to pick restriction enzymes which recognize restriction sites that are not present inside your gene. Otherwise, you're going to cut up your gene. right? So you want to make sure that you can choose ones that are going to not cut up your gene of interest or your fragment of interest. So that's why there are multiple options in these bacterial plasmids that you can purchase. So we have in the bacterial plasmid, we have this particular restriction site, which is shown here in a different color. And it's going to create these two fragments with, in this case, sticky ends. So now we have these sticky ends in which you can basically complementary bind the sticky ends of your gene of interest. So now we have a gene of interest which has been cut with the same restriction enzymes, and they have the same sticky ends. And you can have, you can actually use two different enzymes so that you have one side that was cut with one type of enzyme and the other side cut with another type of enzyme. So it allows you to put the gene of interest in the correct orientation inside the plasmid. You will need to do that if you are trying to express the gene. So if it's an expression vector, you want to put it in the correct direction so that it is synthesized properly. So now we have the gene of interest, so you can see that the two sticky ends are complementary to one another because the same genes were used. And now we have the gaps right there, right? So you can see that the gaps are placed in there, and then DNA ligase will connect those adjacent bases. And now we have a recombinant plasmid. Now this recombinant plasmid can be inserted into bacteria in order to replicate the plasmid. Okay, so this shows those sticky ends again. So you can actually cut your gene of interest out again, right? So you can all right, so let's talk about using restriction enzymes in order to make the recombinant DNA plasmid and how you would detect it. So to confirm your recombinant plasmid, there's a number of ways that you can do that, but ultimately this is the old-fashioned way and the more laborious way. So if you extract the recombinant plasmid out of your bacteria that you have replicated, you cut the fragments or the products again using the same restriction enzyme. So as previously shown on the previous slide, you can see that you can remove that gene of interest again. So you cut the fragments again, and then you detect them on with a process called gel electrophoresis. So we'll talk about that process. So gel electrophoresis involves applying an electric field to a gel-like substance where your DNA is going to be present inside wells. So DNA is negatively charged. So we have, it's like, you know, when you connect your battery, you have the cathode and the anode, or the negative terminal and the positive terminal. So your power source is going to be somewhere in between there. So of course your DNA is negatively charged. You're going to start on the negative side. And when you apply the electric field, the DNA is going to move towards the opposite charge. So it's going to move towards the anode, which is the positive end, or positive cathode of the gel. So the DNA is going to move along inside this gel substance. And over time, all the DNA fragments are going to be separated. So this is the wells right here that are inside the gel. This is where your DNA started. So after applying the electric field for a certain amount of time, the DNA is going to move towards the positive side. And they're going to move in terms of their length of movement, or the distance that they move, is going to be based on the size of the fragment. So you can determine the size of the fragments. So here we have restriction fragments that are of known lengths. Okay, so this is what's called a DNA standard. And this standard allows you to determine the size of the fragment. So let's say this size of fragment represents, I don't know, 1,000 base pairs. So you can say that your DNA fragments right here are 1,000 base pairs in length. And then the further they travel, the smaller they are going to be. So if this is 1,000, this is going to be less than 1,000. And this is going to be even less than that one. And this one will be even less than that one. Okay, so the smaller fragments are going to travel the farthest. Now, this is also semi-quantitative. So here you can see that these these are called bands right here, and these bands represent a fragment of a certain size. Now, it's not just one fragment, there are a lot of fragments. So if you look at the thickness of this band right here, it's a lot thinner than these bands, so it's progressively increasing. So you can see that they're all the same size. However, there's more in these, okay, and it progressively gets less and less and less amounts of those fragments. So that's gel electrophoresis, and gel electrophoresis have been used in DNA sequencing as well. This shows larger fragments of DNA, but the gel electrophoresis used for the, let's say, the chain termination that we discussed in the beginning is going to be it's going to be a larger gel, and it's going to be a lot laborious because you're going to have many different fragments, and so you're going to have many different bands along the length of the gel. And that's what makes it so laborious. It takes a long time to perform the gel electrophoresis. Those have to be performed very slowly and sometimes overnight. This one probably just took an hour or so. Now let's talk about amplifying the DNA, and then we're going to kind of put it together, putting amplification of DNA together with its use in the DNA's use in DNA cloning. So amplification of DNA is typically done by the process called polymerase chain reaction, or PCR. And we're going to show its use in the DNA cloning process. And I've already mentioned how this is done, or at what step it's usually done, but let's look at the process in a little bit more detail. So polymerase chain reaction, or PCR, can produce billions of copies of a particular segment of DNA. So whatever segment that you're interested in, it could be a gene, it could be several genes, it could be just a segment without a gene in it, right? So any piece of DNA you can amplify. Now, the reason why you want to amplify it is because you need enough of it to be able to detect. There are some new methods now that you can actually detect like a single molecule of DNA, but those are very expensive. But in most cases, you want to amplify it so that you have many copies so you can actually detect it using whatever instrument that you have. So PCR involves 3 steps. And the 3 steps are involved in synthesizing DNA. So it's like DNA replication in a tube. So the 3 steps are: 1, first you want to heat the DNA, usually to over 95 degrees Celsius for a certain amount of time. Now this denatures the DNA. And what does that mean? It means you're separating the DNA from double-stranded into single-stranded DNA. And the reason why you want to separate the DNA is just like what happens in DNA replication inside your cell, right? So the strands are separated. They're usually separated by an enzyme called helicase, right? Inside the cell, in vivo. But here we're in vitro, we're inside a tube, and we're not going to put another protein in there, so we just use heat in order to do that. Remember, this is denaturing the DNA. You're separating the strands, so basically you're breaking those hydrogen bonds. And then you want to somewhat cool it. Okay, so you want to cool it down from 95 degrees down to the temperature which is optimal for the primer to bind. So if you remember DNA replication, there's a short segment of DNA called the primer. And the primer allows, basically, it complementary binds to a specific region of your DNA sequence, and it allows the incorporation of additional nucleotides so that you can actually synthesize the DNA. Remember, primers are required in vivo as well, except they're RNA primers, because they're going to be degraded later and then replaced with DNA. However, in vitro, you use a DNA primer. It's more stable, and it stays incorporated into your DNA segment. This is the annealing step. Okay, so annealing means that the primer is going to bind. And then you heat it to, so the temperatures usually change to the extension, and this is going to be the DNA synthesis step. So extension is basically where it's going to be the optimal temperature for DNA polymerase, whatever polymerase that you've put into the tube, to synthesize the DNA using your separated strands as a template. Now, we have, what's key here is heat-stable DNA polymerases. So you can, at this point, you can add everything that you need into the tube and never open the tube again. You can add the nucleotides, you can add the DNA polymerase, it's going to be buffered, right? And then of course you add your template. So the heat stable DNA polymerases allow you to be able to add the polymerase without denaturing it under those very high temperatures. So these are DNA polymerases that have been identified in organisms such as the extremophiles, archaea, that live in extreme environments. So these are DNA polymerases that are very stable under very high temperatures, sometimes over 100 degrees Celsius. But you only have to heat your DNA to about 95 degrees Celsius so they can handle that. And some examples are TAC and PFU polymerase, but there's so many others. Now let's look at this method. Okay, so DNA polymerase. We have a genomic DNA sequence. So this is a DNA sequence that you have, you've extracted. So this DNA sequence is your target sequence, but remember, it's going to be part of a larger chromosome. Okay, so it's going to be basically within the DNA that you extracted from your target organism, right? So your organism of interest, and that could be yourself or somebody else. Now you have the genomic DNA inside the tube with all the necessary components, and you go through this process called denaturation. So you increase the temperature to over 95 degrees Celsius for about 5 to 10 minutes, usually, and that ensures that the DNA strands are separated. The next step is the annealing part. So the temperature is lowered slightly, and it's going to be lowered to an optimal temperature for the primers that the primers prefer to bind to their complementary DNA sequences. So the primers are going to bind. And the primers are going to provide this 3' hydroxyl group, which is going to be required for DNA synthesis to occur. And then the temperature is changed to the preferred temperature for DNA polymerase. So remember, DNA polymerase is going to be an enzyme that actually synthesizes DNA, and this is the extension step. Okay, so this process, this shows right here 1 cycle. Okay, so 1 cycle is going to basically take a target DNA sequence, a single target DNA sequence, and make 2 copies of that particular sequence. Okay, so that's just 2 copies. You need a lot more than that. So 2 cycles are going to add 4. So it's doubled every time, okay? And then you want to increase to here, this shows 3 cycles, and this is going to give you 8. But you want to do about 28 to 30 cycles. So that's going to be, 2 to the 28 copies. Now let's take a look at a video of the polymerase chain reaction, or PCR. And this shows PCR happening at the molecular level. So that kind of describes polymerase chain reaction in a video format. Let's talk about using PCR in a number of different applications. All right, so that was amplifying the DNA. So the polymerase chain reaction is used to amplify the DNA, and what we mean by amplification, again, is to increase the number of copies, and you increase the number of copies up to billions of copies. And you can use it in DNA cloning, but at this point, now you have these copies of DNA. You can actually sequence the DNA if you wanted to, or you can use it in cloning. We're going to talk about how it's used in cloning. So PCR is going to produce a high number of copies of a specific fragment that you're interested in, and we're going to use it for cloning. So the primers can be designed to include restriction sites. Remember, we want to cut the fragments with specific restriction enzymes so that we can insert this DNA fragment into a vector. And this allows the product to be cloned so that it automatically, or copied so that it automatically has these restriction sites. The resulting clones are going to be sequenced and error-free and inserts selected. So basically, you want to confirm that the gene of interest was inserted into your vector, and so you must sequence the DNA to confirm that it's there. Let's look at this process a little bit more in detail. So here we have DNA fragments that are obtained by PCR, and they're colored pinkish. I guess you have dark regions and lighter regions. The dark region is going to be the main part of the gene. The lighter region is going to be where the primers were, which have the restriction sites attached. So these lighter regions are going to be where the restriction sites are. Now you want to cut the PTR product with the same restriction enzymes that you're going to be using, that you actually opened up the cloning vector with. Okay, so you design it so that you use the same enzymes. So you can choose whatever DNA sequence or restriction sites that you add onto your primers when you make the primers, and then you're going to use the restriction enzymes. And here, I'm going to talk about this antibiotic resistance gene in just a moment. But this is your cloning vector, and your cloning vector has been cut with the same restriction enzymes that you cut your insert. And inside the cloning vector, we have what's called an antibiotic resistant gene. So this allows you to select for those vectors that have the insert. And we'll talk about how that's done in just a moment. So you have these two fragments that are cut with the same restriction enzymes, and then you put them together in a buffer, and then you seal them with ligase. Okay, so basically you're going to combine them and then allow them to ligate. And then you're going to insert them into bacteria. Okay, so those bacteria that have the antibiotic resistance gene are able to survive in conditions that has an antibiotic present. So let's say the antibiotic resistance gene is a gene for resistance to ampicillin or penicillin. Okay, ampicillin is a form of like a cousin of penicillin. So let's say we grow the bacteria. Okay, so you insert the bacteria and then you grow the bacteria. Now you want to confirm which bacteria actually have the insert in them. Not all of them are going to have the insert. So then you grow them in conditions in the presence of ampicillin or penicillin, similar. Okay, and then those that are surviving in those conditions are the ones that are assumed to have the plasmid present. And just because they have the plasmid present also doesn't mean that the insert is inside the plasmid, right? So you want to perform DNA sequencing. You're going to extract these plasmids out of the ones that survive, and then you're going to perform DNA sequencing to confirm that your insert is in the plasmid. Once you confirm that your insert is in the plasmid, then you can insert that plasmid and replicate the plasmid and then store it in the bacteria. So now you have many copies of your plasmid.
We can use the techniques of DNA technology to recombine and copy genes. A restriction enzyme is used to cut open a plasmid, a small circular DNA molecule obtained from a bacterium. The plasmid serves as a cloning vector. The restriction enzyme cuts only at a certain DNA base sequence, called a restriction site. The same enzyme is used to cut up DNA obtained from a human cell. The human gene must be cloned to produce copies for use or study. A bacterium is induced to take up the recombinant plasmid from the surrounding solution in a process called transformation. This isn't as clear-cut as it sounds. Some bacteria do not take up the plasmid, and some take up plasmids whose LAC-Z genes lack the inserted human DNA. A bacterium is placed on a growth medium. It replicates the plasmid at the same time as its own DNA. Its descendants form a clone of bacteria that all have copies of the recombinant plasmid. The ampicillin in the medium limits growth only to plasmid-containing cells. But which cells have plasmids with a human gene? Bacteria without human genes have intact LAC-Z genes. This allows them to break down a substance in the medium that stains their colonies blue. Thus, the bacteria with human genes are in the colorless colonies.
Okay, so that summarizes the selection process. And there are different ways to select it. Here we show those that are colored and those that are colorless, but there's multiple ways to select for those bacteria or those colonies that contain bacteria that actually possess your plasmid. Okay, so a quick summary of the amplification of DNA and what we just discussed about gene cloning. So PCR, or this process, was developed by Kary Mullis. He was the one who developed this process, and it's been used across many different fields in biological research and used for genetic engineering. So it's made a huge impact. And these are just some examples of applications in which it's been used. To amplify DNA from a woolly mammoth that was found, which is 40,000 years old, to obtain a DNA fingerprint from small amounts of tissue that might have been left at a crime scene so that we can identify the individual that the tissue came from. We could amplify DNA from single cells, including single embryonic cells. And also we can amplify DNA from viruses in which have infected cells, and ones that are very small and early in the infection process, which normally would be difficult to detect. So let's look at some more details in the next two segments.